Um, I'm, you all know me. Greetings to the Evidence and Reasons Academy crowd. Thank you for joining us. We're privileged to have Dr. Joe Deweese. He's an adjunct professor at Vanderbilt at their School of Medicine and Biochemistry. He's also an associate professor at Freed-Hardeman, and he was previously at Lipscomb. And we're just going to talk about the PsyCalc paper. And that, I'm just going to leave it up, leave the rest to him. So hand it off to you, Joe. Thank you.
Thanks, Al. It's good to be with you today and good to get to present. And thanks to those of you out there who are willing to listen to this. So my name is Joe Deweese. I teach at Freed-Hardeman University, where I serve as a professor of biochemistry and director of undergraduate research. And I'd like to share with you about a software tool that we've been working on. And I had presented this before and had this title, Be Patient and Pay Attention to the details, because it turns out we can learn a lot when we're paying attention. I want to start with kind of the broad problem here, and that is some cancer drugs cause cancer. And that may be shocking to some of you. And let me break that down for just a moment here. I'm not saying that in general cancer treatments cause cancer, but there are a few drugs that we know that as a side effect of the way they work can actually cause DNA damage and potentially cause the development of an additional cancer. For instance, the drug etoposide is a commonly used anti-cancer drug. And one of the effects of etoposide can be not only that it kills cancer cells, but it can damage DNA in healthy cells. And in some cases, a small percentage of cases, can actually lead to the development of a leukemia. So clearly that's not the plan. But the question is, can we make these drugs safer? And in particular, I'm focused on topoisomerase 2, drugs that affect topoisomerase 2. This enzyme untangles DNA during replication, transcription, mitosis, and DNA repair. We know that there are a number of drugs already that are on the market that are used to target topoisomerases, both the type 1 and type 2 that I'm focused on here. There are also, by the way, bacterial versions of topoisomerases that are targeted by antibacterial drugs. So across medicine, this is a pretty important family of enzymes. To break it down here, topoisomerase is a big word, is describing enzymes that change the topology of a molecule. And in this case, these are DNA topoisomerases. And so their role is to change the topology of DNA. You say, well, okay, well, what is topology? Well, we deal with this in life in different ways. So here is a extension cord or the weed eater line or a power cord. These types of things can get twisted up and tangled. I don't use the telephone cord anymore because how many people actually have a phone with a cord? But some of you who are old enough to have grown up like this, like me, you remember the phone in the kitchen that had the cord that went down to the floor and then came back up, and it was always coiled, and eventually you went to answer it and you couldn't pull it away from the wall because you were stuck there. That's a topological problem. Turns out in your cells, you have about 6 feet worth of DNA. And this is true throughout your body. In normal diploid cells, meaning cells that have 2 copies of your genome, it's about 2 meters worth of DNA. And that 2 meters of DNA has to get packed into an incredibly small space, greater than 10,000-fold compaction. So how do we keep that from getting knotted and tangled? Not to mention the fact, too, that not only do we have to keep it from getting all knotted up and tangled, but we also have problems that arise when we process that. So for instance, when we're copying DNA in replication, so here's kind of a simulation of what a replication fork proceeding in this direction would give you 2 sister chromatids on the backside, but look what's happening with those. They're wrapping around each other, becoming intertwined. We call that catenation, or pre-catenanes in this case. And then as the progression of the fork proceeds, you get supercoiling ahead of the fork. We call that positive supercoils, meaning the DNA is actually getting tighter. And the way I illustrate this for my students is if you took a double-stranded rope and you pull it apart, you're going to get a tightening of the compaction of those coils ahead of the replication fork. Same thing in essence happens during transcription. You get positive supercoils in front of transcription, negative supercoils on the backside of transcription. And so what do we do with all of this? Our cells have to alleviate this twisting and turning, or much like the telephone that can't be pulled away from the wall, these processes are going to come to a halt, right? It's going to stop transcription. It's going to stop replication if we can't alleviate these things. We have in our cells enzymes that are designed to do that. And so we have, for instance, DNA topoisomerase 2, which, let me make sure this is, here we go. DNA topoisomerase 2 is depicted by this little molecule coming at us in blue and orange and yellow. This full animation, by the way, is available on YouTube, so you can look that up. Maybe Sal can link that later. So as you see, the DNA gets bound here in the active site. We got two strands, one double helix we're making transparent for the moment here. And notice the lower double helix has been broken in two and connected to the enzyme for the moment. The other double helix gets passed through that break. The enzyme then reseals the break. And there's a little more, we think, structural movement that goes on coincident with that. And then the DNA is going to get released from the bottom. So what in essence has happened right here is you've taken two pieces of DNA that were looped around one another, made a double strand break in one segment, passed the other DNA through that break, and then put the DNA back together. Just a fascinating process and machine. And like I say, you can look the rest of that animation up. This is the way I depict it just as a schematic, where you have here kind of this maroon segment of DNA and a gold segment. Notice we bind to the maroon one and capture the gold one here, and then we'll make a double strand break in the maroon double helix, pass the gold through the break, and then release the gold, and then later release the maroon. And so this progression, this cycle is taking place in our cells all the time to maintain DNA at a proper state of supercoiling that allows for the continued processes of replication, transcription, and other things that are going on. So we need this activity. In fact, without this specific function, our cells would die. They wouldn't be able to survive. You wouldn't get cell division. You wouldn't get completion of DNA replication. And so you'd have massive failures. Turns out topoisomerase 2, there are two of them in humans. So we have actually 6 different topoisomerases, but 2 of them are of this type 2 form. We call them topoisomerase 2 alpha and beta. So I have them abbreviated TOP2A and TOP2B. TOP2A is more involved in replication and mitosis, where TOP2B is more involved in gene expression, chromatin remodeling, kind of housekeeping functions. So in our cell cycle, TOP2B, beta, is expressed constantly. TOP2A rises and falls with replication mitosis. So as you go through S phase and then get over into mitosis, you get kind of a peak of expression, and then it drops off after that. And so you see that kind of cyclical expression with TOP2A. From a drug targeting perspective, it would be ideal if we could target alpha and avoid beta, because it turns out some of the side effects of topoisomerase 2 drugs we think may be mediated by the action of beta and not alpha. But alpha would seem to be the one that would be more important from a cancer perspective, because cancer cells are trying to grow and divide, and maybe more dependent upon alpha. And so we think that maybe if we could selectively target alpha, that could be safer. The problem is the main difference between these two proteins, if we use my little schematic down here, they have the same basic structure through the first part of the protein. About, I would say, roughly 60 to 70% of the protein is really kind of the same machine, if I can call it that. On the back end, we have this gray area called the C-terminal domain, or carboxy terminal domain. It's just the end of the protein. That's all that CTD needs. But it's about 400 amino acids long. And it contains some important components like localization sequences and other things. Well, it turns out that that's the part that's the least similar between alpha and beta. But it also happens to be that that part is intrinsically disordered or unstructured. And what we mean by that is the C-terminus doesn't fold into a nice, neat shape. And so there's an IDR plot there at the bottom showing the black line just past 1,000 up to about 1,500. This region here is unstructured. That happens to be the region where alpha and beta differ the most, right? So they're very similar throughout this part, very different out here. So the question is, what's going on at the tail, and does that help us in any way? So we think it's unstructured. That means X-ray crystallography, cryo-EM really struggle to actually give us any information on that. Sorry, I'm jumping around here. And when we put it in AlphaFold, just the basic AlphaFold version, if I can call it basic, right, gives us this look right here, where the C-terminus is just a squiggly line. Now, that's only half the protein. We're not showing you the other half because it's actually 2 copies together. But you see the yellow, orangish squiggly line running around. That's the gray part in the schematic at the bottom. It just makes a big squiggly line in AlphaFold. AlphaFold doesn't know what to do with it. So my lab specializes in topoisomerases. We can make mutations in them and express and purify them in a yeast system, and then have those purified enzymes to run experiments on. So a few years ago, we set out to make a series of mutants in the C-terminus. So we made mutants across different parts of the C-terminus, focusing at this point on positions that could be modified by phosphorylation. So serines and threonines primarily is what we focused on. And we mutated these serines and threonines with the idea that if we change those sites, could we see any difference in activity in the enzyme? In other words, are any of those sites even important or useful? So you can look that study up. The citations there at the top right, that's an ACS Omega for 2021. Long story short, we found several mutants that when we made those changes, either had slightly decreased or in some cases slightly increased catalytic activities. So that supported the idea that the C-terminus is involved in catalytic activity, not so much as the actual machine, but maybe as it relates to how it interacts with different substrates and maybe helping select which substrates to interact with. And so that was kind of an interesting thing for us to think about, is what's going on, what's the role here that topoisomerase 2 C-terminus is playing. And then again, the overarching theme here would be, if we can understand how the C-terminus works, could we potentially target that area of the protein therapeutically? Since it's so different between alpha and beta, maybe we could make an alpha-specific version, of a drug. So we don't have a structure. We have some mutants that appear to work. And so what can we do to understand more about what's going on in this region that is what we call intrinsically disordered? Now, this is a growing area of study. For those who study proteins, the idea of intrinsic disorder doesn't mean that it's always floppy, but it means that maybe it requires certain other factors for it to fold into a shape. Maybe it's that it binds to DNA. Maybe it's that it binds to protein. Something could be interacting with it. So the question is, can math save the day? A few years ago, I met at a conference Dr. Kirk Durston, who presented on this work that he did a few years back on studying interdependencies within a protein sequence. And it dawned on me that if you can, by way of multiple sequence alignments, analyze a protein and look at what sites are interdependent between one another, maybe that same concept would hold true for the C-terminus, that maybe it might teach us something about what parts of the C-terminus are important or are interacting and so forth. So I got together with Dr. Durston. We gathered several collaborators together, including yours truly, Sal Cordova over there. And we're able to actually, thanks to a really, really talented programming student who's now out in the working world, Thomas Townsley, we were able to take Kirk's concept, which he didn't have as an individual working program, but we're able to take that concept and make it into a working program that you can actually run that we call SciCalc, or Protein Sequence Interdependency Calculator. And with the SciCalc tool then, could we actually analyze these sequences? Let me walk through for just a couple of minutes how this works. You can see the citation there of our paper from 2022. The idea is a multiple sequence alignment is, think of this like a spreadsheet for those of you not maybe in this area of science. Think of it as a spreadsheet where every row is a version of a protein from a different organism. And then every column from columns B on represents sites within that protein, meaning amino acid positions. In biochemistry, we call this the primary structure of a protein, meaning the actual amino acid sequence of the protein. And so here you can see in my example, you've got all these different versions, 1 through 20. And then from columns B on, you've got different positions. Now, in this example, there's no difference between these. These actually happen to be spike protein sequences, I think. And so there is no real difference here because pretty much they're all the same in this region that we're looking at. But let's look at one where maybe it differs some. Here's some different topoisomerase 2 sequences from different organisms. And what we see, at least in the little sample of sequence we're looking at here, is that in some cases, you can match up parts of the sequence that seem to track together. And so the idea here is, notice that in all the P's right here, where it's a P here, it's always a G here, and it's always an L here, and always a K here. So you can kind of see how there are sites that kind of tend to track along with each other. So here's some a series of G's that turn to L's in this position, and K's in this position, and I's in this position, and D's and E's, and so forth. What SciCalc can do is it can computationally look at all of these columns and rows and identify which sites track together, where when one site changes, this position over here changes, and it will cluster those together. And this is an older, kind of an older version of the interface. It still looks mostly like this, but this is just a video I made of kind of walking through the steps of how you would set this up. SciCalc Viewer, for the most part, still looks mostly like this. You upload a file, which would be your multiple sequence alignment, which you have to prepare and align beforehand. And I would recommend, if you're doing this, put your sequence of interest up front, meaning put it at the top. If it's human, if it's some other organism, put that one first, and then put the others below that. Use a program to align it into a FASTA format, and then use a text file to upload it into SciCalc. And then you can use first row mapping, which will allow you to label your MSA in a way that the clusters actually match that first protein that you selected. There are some other options on here where you can change how much insertion data is, meaning if there are gaps in the sequence, do you want to filter those out? Spread is a way of changing how far apart you're comparing columns, if you're comparing them all together or separate. And this version, by the way, is a bit slower. We actually have a version now that's about 4 or 5 times faster than this. And so it starts flying once it gets to some of these stages. Now, this is a small protein here, and this would just take, in the new version, presumably just a few seconds. But as this thing clusters through, it's going to try to cluster all of the attributes together and develop what we call a polygraph tree that we'll see here in just a moment, that represents kind of the output of this particular method. And so there's the polygraph tree popping up here. What's cool about the polygraph tree, and here it is where I kind of show you the manipulation of that. The polygraph tree viewer allows you to actually hover over a site and see what positions cluster at that site. So as the cursor is hovering over these sites, it's giving you an SR mode, which is kind of a measure of the strength of the cluster. And it's giving you in parentheses a series of amino acid residues. And those might be 2, 3, 4, and so forth. And you can actually zoom in on different areas in the viewer and hover over different points and see what positions cluster at that particular node, and then how strong of a cluster that is based on the SR mode value. So that's pretty cool to think about. Go ahead, Sal.
I'm sorry to interrupt, but for also the sake of the viewers.
Yes.
When you're hovering over, so what is the number on the top? You have like 9, 5, 1.
Yeah, these numbers we've actually, I think, gotten rid of. It was kind of keeping up with when some of these things were clustering in the process, and we found those that really wasn't super helpful in the analysis, and so we've actually gotten rid of those. But we have maintained where you can hover over and you can see the actual site numbers and things like that.
So can you hover over one of them and just explain the site?
I'll have to zoom back on the video here because this isn't live. There we go. Here's one. So notice it says cluster, parentheses, and close parentheses, and then it's got a series of numbers, 118, 143, 154, etc. Those would represent different amino acids in this particular multiple sequence alignment that have clustered together. The SR mode would represent the strength of that cluster. It's a 0.12. And for a cluster of 8, that's actually not terribly bad. I think that's 8 sites. So that's actually a pretty decent SR mode, probably a fairly strong cluster in other words. The highest would be for a cluster of 8. I'd have to do the math on that, but it's not, let me see here. Trying to do it off the top of my head here. Yeah, it'd be like 0.25 would be the highest you could get for the SR mode for eight sites. So it's not terrible. If you have just 2 sites, like this one up here in red, is a SR mode close to 1, and it has just 2 sites. 1 would be the highest you can get with 2 sites.
Oh, so those are the number of amino acid positions that are in that cluster that are.
Yeah, over here on the left, this represents the number of positions in that cluster. So these all have 2, 3, 4, 5, 6, 7, so forth. So down here, it skips because some clusters may not have a number, but this one has 21 at this point, 22 here, and so forth.
So if I could just interject here, the one thing I do remember is when Kurt ran this in his peer-reviewed paper like 12 years ago, he was able to detect van der Waals interactions at positions. That was, to me, that, that really leaped out. That was just like, wow, that's kind of hard not, it's kind of nice to know you could just look at the sequences across species and it's going to give you that. That was when everyone was kind of getting, his dissertation committee was like, wow, that's really cool.
Yeah, it's pretty impressive. And to Kirk's credit, he used a really diverse sample set. We'll talk about in a minute the problems that come when your sample set is not, let's say, as diverse. But he was able to, ubiquitin is present in a lot of organisms, so he was able to get a really broad sampling, which I think really strengthened his results. Yeah, it's a good point. Absolutely. So this mechanism or this approach then can allow you to cluster these things together. And I'll show you in a few minutes what the newer output looks like, because we actually can output to a spreadsheet and get a ton of information out of it. So what can we do with these sites and these clusters? Well, we can actually map them back onto structures. And so here's an example from our 2022 paper, where we mapped those sites back onto topoisomerase II and demonstrated that these different clusters represent, things that are interdependent. Now, I'm specifically using the word interdependent because our tendency is to say, well, that means they interact, and they may not actually physically interact, meaning sometimes these interdependencies can be quite distant. And so we don't necessarily understand the nature of how those interdependencies come about, but we understand that they are there. Sometimes it appears, I would say, I think we have decent evidence that it appears we see a lot of this at the ends or the anchor points of, let's say, helices and sheets, which makes sense. That's a stability point when we study that in biochemistry. So that's something that may be happening in those, but we can study these on different levels. We can look at different domains. Here's another example where we pulled out a data set from looking at the ATPase domain of Topo2, and I tried to color them in all these nice different colors just to get an idea of that these interdependencies could be all over in different places here. And even though 2 sites may be next to each other, they may not necessarily cluster together. They may cluster with something a little bit further away, which is not necessarily intuitive in all cases, right? In other words, this is showing us interactions that are a little harder to predict. And even between domains, so here's kind of getting it back to that point. Here's examples of clusters where you see they're color-coded. So you see the yellow, there's some yellow up at the top, some down toward the middle domain interacting with the DNA. There's some kind of that kind of cobalt blue look going on in there. And so some different examples where you see these sites can be spread across different areas of the protein. Now, obviously, we got these clusters, right? So then what, right? Can we use these clusters to understand anything about the C-terminus? So as I mentioned, we made these mutations previously with the serines and alanines, mutating them mostly to amino acids, serines and threonines, mutating them to alanine and glycine. Some were catalytically unchanged. Most actually changed activity a little bit. And so this was a little bit of a paradigm shift because in my field, the thought was small changes in the CTD really wouldn't alter activity. But we were able to demonstrate convincingly that you can use these small changes and get differences in activity. So can SciCalc give us some clue as to what's going on? So we ran some analyses, and in our 2022 paper, we published a series of clusters. And here's an example of some of those. And so we made some mutations based on the clusters that we found. So here's 1317, clustering with some residues, 1462, 63, 82. These higher numbers here represent C-terminal domain positions. These lower numbers represent positions up in the ATPase domain. So that was really cool, because here we got something way out in the C-terminus clustering with the ATPase domain. And so maybe we can see what's going on here. So this is kind of the, see if I can get that animation to work. I don't know if it's going to work. But you can kind of see that the color coding of my little structure here on the right shows you that you got positions up at the top, and then you got positions along the squiggly line, the C-terminus, which would represent these higher numbers here, the 13, 14, 15. Those positions are represented by that. So as we can see from the structure here, these sites up at the top part in the different colors are interdependent with sites out in the squiggly part. At least that's what the evidence appeared to indicate. We made a series of mutants. I had some students pursue experiments with those mutants and do everything from what we call relaxation assays, DNA cleavage assays, binding assays, decatenation assays. And so we gathered a bunch of data from those. In the meantime, I was made aware of another data set of TOPO2 sequences, 105 TOPO2 alpha sequences that were all cleaned up. And I got really excited about that. And I said, I want to run this through. So while my students are doing their work, I'm working on this, trying to study these 105 TOPO2 alpha specific sequences to see what can I learn about it. And so I got these neat clusters. So here's an example, by the way, of the progression of perfect clusters. So these are maximum SR mode values. So at a 2-position cluster, you can have a max of 1, then 0.67, 0.5, and so forth. And so you can kind of see where the maximum SR modes would be for each of those levels. We, in the meantime, did some additional work on this. We're thinking about clustering and visualizing data. And one of the things we did in this time is we said, well, what if we take those clustered positions and group them together on a spreadsheet and look and see if we can see the patterns like the program is. And this is one of the clusters that we got. And so you look at it, it's a 0.25 8-position cluster. So it's a perfect cluster. But what looks funny about it? If you notice this column, all those columns essentially don't change. And then you get to the last column and you get all kinds of change. So how on earth are these, how on earth is this a perfect cluster? And so that got us asking the question, wait a second, is this a general problem or a one-off issue? So we went back and we looked at previous data, including the data from which we designed our mutants. And this is really humbling, by the way. And what we found is that all of these sites that we thought were clustered appropriately were not. At least the ones that we were looking at for our mutants. Okay, a lot of other sites were just fine. These sites, though, that I'd picked for exactly the ones I picked for the mutants, right? This is humble pie in action right here, where I had to take this and say, wow, okay, these are not what we thought they were. Because what we found is that the positions with the lower numbers, like 62, 63, 73, were probably appropriately clustered. If you looked at the whole alignment, that was probably real. But the last position was thrown in there, we think, aberrantly. And we figured out in the software what was going on is that sites that have little or no variation are really hard for the program to cluster. In other words, if you got these sites here and they don't ever change, then there's no way for the program to differentiate what sites have what kind of preference or order and how they should fit together. So the idea was brought to the table to use low entropy filtering. In other words, filter out sites that have little or no change. So entropy in this sense is not a thermodynamic quantity, but an information quantity. So in other words, if there's a site, a column in a multiple sequence alignment that doesn't change, like one of these, where it's just E all the way down, that's a position of very low entropy. and also essentially, kind of tells us something about that site, right? There's not a whole lot of change there, which tells us that site's actually really important, by the way, because otherwise it might have much more variability. But we were able then to apply entropy filtering to our data and end up with stronger correlations within the clusters and avoid these positions where there was little to no change. So that work came to fruition over the past several months and resulted in a publication just a few months ago with a couple of my students there, Clark and Corey. And one of the things that we found, here's an example of a cluster that we got, and Clark, in a very fun manner, was able to actually color code this for me, because I thought we need to be able to illustrate this. I hope you can see by looking at the color coding that the color changes seem to correspond between these positions. Not perfectly, okay? There's some positions that don't match perfectly, but there's a number of these sites here where the correspondence is very, very high. And if you look across the top, those numbers represent the position of those amino acids in the actual sequence. So you got things at 100, 300, 500, 600, 1100, 1400. So some of these sites are far apart, very far apart, but yet have really strong correlation. And I'm not showing the whole alignment here. This is just a sampling of the organisms that we were looking at for this particular example. But you can see that what we see in the lower right here is the actual entropy value by site throughout the protein. So topo2 alpha has 1,500 amino acids. And there's 2 copies of that come together to make the functional protein. But if we just look at the sequence in and of itself, it's 1,531 amino acids. And what you see on the little chart there is for the first, about 1,000 amino acids, there's some spikes, but a lot of it is down there below a threshold level. And I set a threshold example of 0.1 entropy there at the bottom. And as you get out to past 1,000, what you notice is the trend is for those sites to have a much higher entropy, and meaning that those sites change a lot more. Okay, they have a lot more change among those positions in those sites. And it turns out that those are much easier to cluster than sites that don't change, right? And so we're able to actually filter that out. And using the latest versions, we can actually output this to Excel. And here's an example of what the Excel sheet looks like. And I've clicked on the 4th order tab here, so you can see what 4th order clusters look like in this particular data set. Now, we're not seeing all the positions. This is cropped, but you get a kind of a sampling here. And we went ahead and created tabs that go up to about 10th order. And we also created a tab that tells you what were the low entropy positions that were filtered out. So you can actually, in the data set, look at which positions did we just eliminate and avoid clustering for. It turns out with topo2, this is really fascinating. With topo2 alpha, we actually cut out a large number of sites, and it's close to half the protein doesn't change, which is really fascinating. Even across 100 different sequences that were used in this particular analysis, 105 different topo2 alpha sequences. We've scaled that up. We have over 450 some now. I don't have this data on here to show you at the moment, but even among about 450 plus sequences across chordates, we see that stays the same. About half the protein doesn't change. What that tells us, though, is those low entropy sites are really, really important. Okay, so understand that. We're not saying they're unimportant. We're just saying we can't cluster them. But then we can cluster the others and come to some understanding of what's going on in those other sites. And so you can see here through the clustering, we can actually take that, map it onto the structure. You see this particular cluster is all over the place. It doesn't have one particular site. It's C-terminus, it's amino terminus, it's central, all over the place. So, what do we get from this, or what's kind of the takeaway here? I think what I would say from this is that SciCalc identifies these interdependencies in amino acid sequences. Low entropy positions can cluster anomalously with a high entropy position. And so this entropy filtering seems to eliminate that. SciCalc now outputs these clusters into a spreadsheet where you can actually look at them from second to 10th order. You can get all of the data, even higher order clusters beyond that, but we have tabs at least up to the 10th order automatically generated for you. And so you can view those clusters, you can look at the correlations, you can look at the SR mode strengths of those, that's on the first tab of the results, and get some idea of how these different positions are working together. So it's really fascinating stuff. Certainly got a number of collaborators here. Thomas Townsley, a friend of mine now, Mark Soda, has done a lot of updates to the program for us, along with, of course, Kirk, Dr. Tim Wallace over at Meharry, and Sal. And then we've had a number of students like Clark Inslee and Corey Moore and Matthew Cool who've helped with this. So one of the questions that I want to mention here before I close that we've tried to get to is, all right, if SciCalc can detect interdependencies within a sequence, is there any reason why you couldn't take two sequences and detect interdependencies between those two? And the answer is no, there's no reason you couldn't. And we actually now have a version of SciCalc. The interface is just a little bit different from what we were seeing earlier. And this new interface will allow you to bring in more than one protein MSA into the program, and it will not only look within that MSA, but between two or more MSAs. In fact, you could put several in there if you wanted to compare a bunch together all at once. It will and can computationally do that. And examine and identify interdependencies. Now, the caveat again is if you don't have a lot of variation in those sites, they may get filtered out. And so some of these sites, if they don't change, may not be visible with this method. So that's a caveat, but I do still think there's some real strengths here in terms of how this works. So kind of where we're headed next is we have some new alignments that we're building. We have several proteins that we're going to look at and potentially look for interdependencies between proteins to confirm that we can actually see that and see if we can validate that, because this could become a pretty cool prediction tool that researchers could use to compare or to analyze MSAs from separate proteins and see if there's any possible interdependencies, which could represent binding sites between those two proteins, which would be a really neat thing if we could predict things like that. So we'll see where this goes, but I'm really excited about it. The work has been great. We've been incredibly blessed to be able to work on these things. I got a great crew of folks here at Freed-Hardeman doing work with me. And so we have a lot of fun doing what we're doing. So with that, Sal, thanks for having me. I appreciate the time to present, and I'll see if you have any follow-up questions before we close out.
I have two comments. SciCalc is not isolated in this approach. There's a very well-developed community, direct coupling analysis.
Yes,
And
absolutely.
this whole thing about low entropy and high entropy sites, that's also very important to that community. So we basically have to filter out the low entropy sites. We would call in evolutionary terms conserved area, conserved sites. And so I'm sure our, the SciCalc community is not alone in the challenge of having to filter this out.
Absolutely right. Absolutely
but
right.
the DCA community is very well developed. They've done fabulous things as part of AlphaFold. The second thing that I'll mention is the real direction that I feel this could be so powerful is what you said at the very end, when you're comparing two protein sequences, or even hypothetically protein and DNA sequences, because there's a lot of transcription factors that bind to DNA. And there's a lot of variation there. We could now probably predict where the contact sites are in interactions, because we are trying to build designer transcription factors with zinc finger proteins. We don't know what we're doing very well. The designs that exist work very well. If we can build, we're trying to copy the designs that already exist, we could probably get insights using this tool. So that's where I've always seen where the future could really be. It could be there, and it's really exciting. So, I just
makes me wonder if there's a machine learning approach to train data sets between proteins and DNA sequences to find, can that be honed in where you could predict that if you had the protein sequence, you could predict where it binds. So that'd be kind of cool.
Oh, absolutely. But, we're still we don't quite have enough data, across species. We know that there's covariation across species for both like the DNA and the protein, but we don't quite have anywhere near 400 data sets to be able to do what we just, even 100, just like what you did with, one protein in isolation. But it'd be nice to have the tool in place to do it when that data comes in. So that's all I had. By the way, I didn't realize your team was that large. Wow.
Well, so a lot of those are undergrads here at Fried-Hardeman. They're great students. Some of those just graduated, so that was kind of one group kind of moving on and another group coming in. But they some of them are off in med school, pharmacy school, graduate school, doing different things now. So yeah, it's just been tremendously blessed to get to work with some great folks. And some of these students, by the way, also work with other faculty. So, it's not always, hey, just work with this professor. Some of them do research in a couple different areas, which is cool to see too. So,
well, I just have to say it was a real honor to have a chance to be a co-author with some of your students. That I can't tell you how much that meant to me. So thank you, Joe.
Absolutely.
That's all I had. This is.
That's all, folks.